for organizing such a great conference. So, as Dave mentioned, I am speaking about magic populations next, and in particular, map construction in magic populations. So this slide, it shows a diagram which we've already seen before, just highlighting the fact that in crop genomics, one of the biggest challenges is really the fact that we have complex genomes. So a lot of people think of the human genome as the standard, but when you look at crops, there's many which are much larger than the human genome, and the one which I work on in particular, wheat, is much, much larger. It's hexaploid, there's lots of redundant sequence. And as Neil Stein pointed out yesterday when talking about barley, what this really means is that we need better resolution, higher density maps. And so he talked about one route to achieving that, but today I'm going to talk about the way that we've gone about achieving these types of maps, and that's by using these magic populations. So magic populations have been developed now in a number of model organisms, mouse, Arabidopsis, Drosophila, and then a number of crops. So on the left here I have rice, I have wheat, I have maize, but they've been developed in durum wheat, in tomato, in oats, and in chickpea and pigeon pea here at ICRISAT. And they're growing more and more of them every year, actually. And the reason behind this is, as Dave pointed out, so I don't have to go into this slide in too much detail, just like NAM, they occupy a niche intermediate to biparental populations and association mapping populations, both in terms of the amount of power you have for QTL mapping and other trait mapping approaches, and in terms of the diversity that's brought in through the parents. Whether you have 4 parents or 8 parents or 16, it may not be as much as a NAM population, but it's still more than a biparental. But all of this relies really on having a good linkage map. So this is to give you an idea of what a magic population looks like. These are the schematics of the pedigrees for the 4-parent and 8-parent populations that we have at CSIRO. So on the right is the 4-parent population, and basically we're just intercrossing the parents until we get down to a stage where we have individuals whose genomes are made up of parts of all of the 4 different parents, and then we create inbred lines from those. On the left, we have the 8-parent population, which has 3 parents in common with the 4-parent population. And again, we're breeding the parents together. And then we actually divided the population into subpopulations. So a large subset of them we just create inbred lines from. And then 2 other subpopulations, we actually have additional generations of intercrossing to see how much additional recombination we can introduce and what effect it has on our ability to do genetic mapping and trait mapping. So the 4-parent population is really aimed at the Australian wheat industry. The 4 parents represent different wheat growing regions in Australia, and we have about 1,500 F6 progeny lines. And then the 8-parent population has 3 of those Australian parents and 1 each from the US, Canada, China, Mexico, and Israel. And that's much larger, actually. We have almost 5,000 of the lines which just go directly to inbreeding, and then 1,000 of different subpopulations. And we've genotyped about 3,500 lines from all of these. So it provides really a great resource population for doing all sorts of analysis. Now, the advantages of the magic populations are that we have more genetic diversity, we undergo more generations of recombination, and both of these, in addition to the size of the populations, makes it a great platform for linkage map construction. As well, the genetic diversity has led to great phenotypic diversity. So the populations were originally devised for looking at quality, but we found that basically every trait we look at in both the 4-way and the 8-way is segregating. So that includes traits related to disease resistance, to nutrient use efficiency, to yield, and so on. And so all of this means that we can use the resource that we've built to look at a number of traits, a number of different environments, and investigate really what's going on in the controlled structure of the population. There are a number of challenges that go along with this, though. So the main one is that we have biallelic markers in a multi-parental system, so we don't know exactly which alleles are coming from which parent, and furthermore, we're only genotyping the parents and the progeny, so we can't directly track the inheritance of alleles. The size of the population and dealing with these additional challenges mean that there's a higher computational burden for genetic analysis, and of course, trying to phenotype 3,500 lines means a much higher phenotyping cost. So there are a number of opportunities for genetic analysis with these populations. QTL mapping, looking at genomic selection, and looking at more advanced opportunities such as G/E interactions, multi-trait, and epistasis. But all of these really rely on the foundation, which is the genetic map, and so that's what I'm going to focus on today. So the genetic map is, the motivation from that comes mainly from the fact that we'd like to have a high-density map to anchor the physical map to produce a high-quality reference sequence. And so this is a diagram from the International Wheat Genome Sequencing Consortium website. It's much more complete than it was when I first started working on this, but you can see that we're still a long way from having a reference sequence. The only chromosome that's completed is 3b, published last year, although we're getting close with several other chromosomes. We do have physical maps, though, for almost all of the other chromosomes, so it's really an ideal time to be producing these high-density, high-resolution maps. Of course, another motivation is the fact that we have greatly increasing genotyping capability in wheat. So in 2011, the most dense maps really contained about 3,000 to 4,000 SSRs, DARTs, and RFLPs, whereas in 2012, not only was the 9K SNP chip produced and mapped, but Jesse Poland and Mark Threll showed that you can map GBS markers and produced a 20,000 marker map. And then more recently, there's the 90K chip, which is out, the 400K chip, and GBS is certainly becoming more and more popular. So being able to map where these markers are in the genome really gives us opportunities that we haven't had before. So I'm going to talk about some of the problems that go into actually constructing genetic maps in MAGIC populations and how we went about overcoming them, and then finally show you some of the results from these populations. Now, the most basic problem that I mentioned before is that we do have unobserved information. So we have biallelic markers, we have multiple parents, and we can't track which alleles are being inherited from the parents down to the progeny. And that means that we can't actually track exactly where recombination events are occurring without having some additional information. Now, luckily, before I started work in this area, Carl Bowman had also looked at this and had derived probabilities for inheriting these different alleles, not for the purpose of map construction, but for actually doing QTL mapping. But we can use these probabilities in estimating recombination fractions and then use those to create maps. Of course, this does result in a more complex analysis than you would have in a biparental population where you can see the recombination events directly. And what that means when we're looking at these high-density maps and trying to map 90,000 markers is that there's a much higher computational burden. Now, the way we've gone about this is moving from R code to C code and eventually to using graphics processing units or massively parallel processing. And these have been of great help as we've increased the number of markers that we're dealing with. So for small-scale examples, we've been able to reduce the time used from about 2 hours down to 20 seconds. And obviously, as you increase the number of markers, that becomes a big advantage. So those are the two main issues we had to deal with in actually creating genetic maps. But that doesn't really capture the scope of the issues in MAGIC populations. So even once you have a draft map, there's a number of questions you want to answer, and there's a number of ways you can improve it. So one of those is the resolution limit of the map. Now, when we're talking about GBS or 90,000 markers, we're really trying to saturate the genome here, and even with 8 parents and a number of generations of recombination, we're not going to be able to resolve all the markers. So we've taken the approach of using bins where we put together markers which we don't observe recombination between. And the advantage this actually gives us in MAGIC populations is that we're moving from a number of biallelic markers which can't distinguish between all the parents to haplotypes which can distinguish between more of them. So we're moving to more informative markers. So over here on the left, we might have 3 markers which have no recombination between them, and they're all biallelic, but they represent 3 different haplotypes. And by converting these to a bin and using these multi-allelic haplotypes, we actually are getting more information from our data, and that not only helps with the genetic map construction, but also will help in QTL mapping to give us more power. Another question that we've addressed is looking at the issue of quantifying map uncertainty. So even if you produce a map, you have no idea how good it is. What other orders might be just as good? And one of the issues is that the probability of getting the correct ordering depends not only on the marker spacing and the sample size, which you would expect, but also on how the different alleles are distributed among the founders. So if you have more informative markers, which are represented by the blue line, then even for smaller sample sizes and smaller marker spacings, you do a good job pretty much every time. But if you have different combinations, you can end up having a much lower probability of getting the correct ordering. And so on the right here, looking at different marker spacings, it's not really until you get to marker spacings of 2 or 3 centimorgans that you can be completely sure of ordering all the markers correctly. Although that's still pretty good when you think about these populations. So one of my students, Daniel Afak, actually developed a hypothesis test to more quantitatively look at the evidence for different orders across the genome. And although he developed it specifically for multiparental populations, it actually works generally, so it can be used for looking at map uncertainty in other populations as well. And we've applied this to chromosome 3b, so looking at triplets of markers across the genome and quantifying the map uncertainty. And then compared it to the physical map by actually sequence mapping the markers to the pseudomolecule. And you can see there's regions in the middle which are red, so they're more uncertain, and that's because we have a large number of markers in those locations. They're much closer together. It's harder to tell if we have the correct order. We actually have a number of markers up in the corners which are saying that they're not mapping to the same position in the physical map as they are in the genetic map. But his method actually calls those as quite certain. We actually think that those are in the correct location based on our data. And when we compare it to other biparental maps, these markers are actually mapped to corresponding positions in the biparental populations as well. So in some cases, this can be used to help identify issues where there may be differences from the populations used for the physical map or actually errors in the physical map. And then one of the other problems is the issue of segregation distortion. So in our population, we know that there's a large region on chromosome 2b representing the SR36 introgression, which distorts segregation and likewise distorts recombination fractions in the vicinity. And so another student, Rowan Shaw, actually came up with a correction to reduce the bias in estimation by accounting for this distortion. Normally, you would just get rid of all the markers which had very high segregation distortion, but this would mean throwing out half the chromosome in our case. And so if we can include those markers, we can actually get a better idea of what's going on in that region. So this relies on the assumption that we know there's a segregation distortion locus, or SDL, and that we have dense enough marker coverage that we can have a marker close to the SDL. Both of which are very true in our case and true in many other cases. And then we can use the fact that we have such a large population, which is derived from many different combinations of mixing up the parents, to get an approximate correction for the distortion by considering the effect of the segregation distortion locus in different subgroups. So when we apply this to chromosome 2b, we can see that the green line and the green points, which are the unadjusted positions on the map, this represents the smooth low S-curve of the positions on the map, and you can see that there's this hump in the middle, basically, which is caused by a number of markers which we can't place accurately. So we're comparing the magic map here to the synthetic Apata map, which has no segregation distorter. Once we apply the correction, though, which are the red points and the red line, we get a much more linear relationship, and we're able to more accurately position the markers which are near that segregation distorter. Now, there's a number of additional issues which I haven't gone into. One of those is data consistency. So we are genotyping across many different platforms. We started off with DARTs and some SSRs, and then we had SNPs, and now we have GBS data on several chromosomes. And of course, we've actually genotyped different lines on different platforms, which means there's quite a bit of missing data for each of these cases, and we have to consider genotype imputation. And we actually also have missing data when we're looking at the recombination fractions, because we end up with some non-identifiability issues. But I guess overall the message is we've been able to overcome these issues. So in spite of the fact that magic map construction is challenging, you can end up with some quite good results from it. One of the things that we've been able to do, now that we have a map, is to look at the recombination landscape. So on the left is results from a simulation looking at how many recombination events you expect to see in generating a magic population with 4 parents, which is the blue line, with 8 parents, which is the black, and then compared to a biparental population on the left in red. And on the right are the number of recombination events that we observe in our 4-way and 8-way populations. Now, I'm not sure how clear the numbers are, but you can see that they're actually a bit less than what we see in the simulation. And this actually makes sense when we think about it, because the simulation assumed that the A, B, and D genomes were all equivalent, but we know that the D genome is less polymorphic, and we're not actually observing many of the recombination events that are occurring there, potentially. And when you look at the numbers, it's actually reduced by about a third relative to what we see in the simulation. So at least it's matching up generally well. What this also allows us to do is to track those recombination events and actually try and map QTL for recombination. Now, this is an interesting situation because it's a phenotype that we have on 3,500 lines because we've genotyped 3,500 lines. So we have much more power to detect QTL than you would have in a typical Ville population where you might look at a similar thing. And I'm not going to show the results today, but we have been able to map some QTL basically due to this quite increased power in the population. So the resolution, in the previous slide, I was showing you the number of recombination events, and you can see that we do expect to have higher resolution because there's more recombination. And we have seen this throughout many of the different maps that we've created. So for the 9K map, when we looked at some biparental populations, there were about 817 unique locations, 817 bins. In the 4-way population, there were double that number of bins, and we were able to map about 4,400 SNPs. And in the 8-way map, there were about 2,600 bins, and we mapped about 6,000 SNPs. And then a consensus map was actually created from 6 different biparental populations and the 4-way population, and that had about 2,800 bins and about 6,800 SNPs in it. So it was actually those 7 populations together gave about the same number of SNPs mapped and the same level of resolution as the map created just from the 8-way population. Similarly, in the 90K map that was created, there was a consensus map generated from 6 biparental populations, and that mapped about 40,000 SNPs with about 5,500 bins. And then in the 4-way and 8-way populations, we actually didn't have as many SNPs called to begin with, but we mapped quite a large proportion of them in both cases and had very high resolution. So the last thing I was going to mention is to come back to where I started at, which was using the map to anchor the physical map. And this is one thing we're working on with Rudy Appel's on chromosome 7A, actually, with the idea being to map the marker sequences to different back pools and to look at them, look where they are in the magic maps and look at the recombination fractions between them. So this gives us opportunities both to improve the magic map and to identify issues in the assembly or structural variation. So an example is on the right. So these are markers which were in a single back pool, so they should be, basically, there should be no recombination between them at all, which would be indicated by red. And what we found in the magic population is actually that several of them seem to be linked separately from the other markers in it. So this may either be an issue in the assembly at the moment, or it actually could just be differences between the magic populations and the Chinese spring. And then on the left is actually a comparison of all of the GBS markers and the various pools, the markers that were mapped to pools in the magic population and where they were mapping in the Chinese spring Renom map. So we get very good agreement in terms of the maps that are being used, but we do have more markers that are being mapped in the magic population. So it works very well to help with this sort of analysis. So just to finish up, multiparental populations really do offer novel opportunities in linkage map construction due to the fact that they have many recombination events leading to high resolution. They have many polymorphic markers, so you have very high density maps you can generate. And because we have these large and diverse populations, we have a great ability to map segregation disorder loci. The challenges associated with these really require the development, though, of efficient statistical and computational tools. Dealing with any sort of large data like this would require this. And the tool that we use is an in-house R package developed called NPMap, which has been extended multiple times to deal with many of these issues that have come up and to actually allow for more interactive genetic mapping as well. So I just want to thank my collaborators. So Colin Kavanagh and Matthew Morrell really were the creators of the magic populations at CSIRO in the agricultural flagship. Stuart Stephen works on the bioinformatics side, and Matthew Hayden's generated a lot of the data. I mentioned Rudy and Gabriel previously working on chromosome 7A, and then Rowan, Andrew, and Daniel work within the digital productivity flagship in CSIRO as well. Thank you.
Thank you, Emma. You finished. We have 9 minutes with us in this talk. So there can be many questions.
Emma, you talked about recombination as a trait of 3,500 individuals. So I would have thought that recombination is actually, the trait of an individual is the recombination events in its progeny rather than the recombination events in itself? Or is there something because it's an inbred line that you can get around that?
So the trait we're looking at is the recombination events in the progeny. So I mean, it's all derived originally from 8 parents. So these are the recombination events that have accumulated since those parents were crossed, although obviously you're not going to be observing all of them in the genome. So it's not the most accurate way to measure recombination, but in some ways it's the best we can do.
Thank you for that very thoughtful presentation. I wonder if you might comment on the distribution of recombination in wheat. If I remember correctly, it tends to be have recombination events located in a sort of telomeric region. So do you think there would be value in the generation of magic populations to select rare recombination events in early generations so that you have a better distribution of recombination events?
Yeah, it's, certainly you do see fewer recombination events near the centromere. That is something that's come up in more than one discussion with other people. So looking at the Arabidopsis magic population, we've talked about the potential of actually locating recombination events in regions of interest and really trying to use those more effectively in fine mapping. So certainly with the large number of lines that have been generated, it gives you many opportunities to do things like that.
Any more questions?
So in a magic population, you would be able to do that, identify recombination in the early stages of the crossing, so you could choose which parents in order to use to construct the magic population. At the
moment we can't, because we're not doing genotyping early enough on, so the population was generated quite a while ago at this point, but in theory, if you were doing genotyping more frequently, then you could do something like that. Yeah.
Yes. Dr. Balyan. Dr. Balyan.
While preparing the magic population, when you make intercross between four-way hybrid, how do you decide the number of plants which are to be intercrossed?
Sorry, how do you decide which parents to intercross?
Number of plants which are to be intercrossed. See, in a four-way hybrid, the hybrid will be segregating. So you'll be having in 8-parent magic population, you'll be having 2 four-way hybrids intercrossed, right?
In the 8-parent, yes.
So how do you decide the number of plants? How many plants are to be intercrossed to generate a 8-parent magic population?
Yeah, so deciding on the number of parents and deciding which parents is, as you may realize, one of the most crucial steps, and that's one of the things Dave commented on as well. So depending on whether you're focusing on diversity or whether you're focusing on material which is more relevant to breeders, you're going to end up with a very different population at the end. The way that we've taken to doing the intercrossing is basically looking at as many different permutations as possible. That's probably not necessary and takes more time than, the benefit you get out of it. But it does mean that, for example, in the case of the segregation distortion correction, you're sort of averaging effects over all of these different permutations, which allows you to correct for things. Whereas if you had a smaller sample of the different combinations of the parents, you wouldn't be able to do that as well.
Okay. Please join me to thank Emma for her excellent presentation.